In 1977, two papers were published which changed our understanding of the expression of genetic information. So those papers describe what the researcher called at that time an amazing sequence arrangement in adenoviral genes. And the reason why it was amazing is because it was going against everything we knew about expression of genetic information at that time. From what Francis Crick used to call the central dogma in molecular biology, we knew that genetic information is stored in the DNA, which is then transcribed into RNA molecules which have multiple functions. And those RNA molecules are then translated into proteins, and proteins are building blocks of our bodies. So our bodies are almost entirely made of proteins. Now, it was known that even though the alphabets of those different molecules are different, however, the information which is written in them is the same. So there's really the linear correspondence between the three molecules, as you see. However, what people discovered in some of the higher eukaryotes is the fact that actually they couldn't find the corresponding sequences that would correspond to the DNA on the protein level. So some parts of the genetic information was missing in the genes. And although initially it was thought to be perhaps just like a very peculiar example of just one case, however, very, very soon it became clear that this is the case for most of the eukaryotic genes. And for those of you who don't know what the eukaryotic genes are, so these are eukaryotic cells are the cells like what we have in our body. So they contain various different organelles, as opposed to the prokaryotic cells, which are characteristic of bacteria, for example. So it became clear that it was a widespread phenomenon, and that most of the eukaryotic genes contains those segments which are not present in proteins. And those segments were then named introns. They are non-coding, and they have to be removed during this expression of genetic information. So as in a simplistic term, we can say that this is the message, this is what we want to keep, and this is what we want to get rid of. Now, very, very fundamental question is, why is it like that? I mean, why would we have pieces of the genes which are non-coding and are completely useless? And in order to understand this question, we really have to look at the evolution a bit. So this is a very simplified evolutionary tree. And then we need to go back to the origins of the eukaryotes. So as I mentioned, eukaryotic cells are the cells which are characteristic for human, for example, as opposed to bacteria, which don't have organelles. And the eukaryotes were actually formed about 2 billion years ago, so very, very long time ago. And the way eukaryotes were created, it is believed that they were created by so-called endosymbiosis. So it was a process during which proto-eukaryotic cell was invaded by the bacteria, which gave rise to the mitochondria, so the organelle which is producing energy in our cells. And in case of plants, there was also photosynthetic bacterium which entered the eukaryotic cells, and that gave rise to the chloroplast. So this is the theory which explains how eukaryotic cells were created. And of course, they had many very different advantages over prokaryotic cells in terms of diversity of the environments it could live in. In any case, so it was very beneficial for the cell that it compartmentalized very specialized processes inside the cell. However, it came with one very, very big consequence. And that consequence actually can be traced down to this very nice molecule which is known as group 2 intron. So group 2 introns are self-splicing RNAs, but they have very peculiar properties. So we know that they reside in the bacterial genomes. So these are the bacteria which actually gave rise to mitochondria. And what we know is that they can be transcribed, and then they can be self-spliced. But they have this property, so this is not particularly important for our consideration, but what is important is the fact that they can reverse splice into genome again, and they can essentially multiply themselves. And that wouldn't be a big problem, because in this particular environment, they can actually self-splice again, so that's not a big deal. However, when the eukaryotic cell was created, there was another genome of the proto-eukaryotic cell, which was present there. And in this case, those self-splicing and mobile genetic elements invaded the genome of the proto-eukaryotic cell. And in this case, the environment was slightly different, and most likely those self-splicing elements could not cut themselves out very easily. and the cell was in a big trouble, so to say, because the whole genome and genetic information got punctuated with the things which could not be removed. So therefore there was a very high evolutionary pressure to evolve a machinery that would be able to remove them from the cells at that time. So if you look at the architectures of the genes, as we know in these days from human cells, you can see that actually those coding regions, which I was talking about before, they don't really constitute a large portion of the eukaryotic genes. So in this case, you can see introns, the non-coding parts, they spread and they were kind of enlarged, and they constitute most of the genetic information, which is actually not used later on. So this looks like a terribly inefficient thing, and obviously it's just a consequence of the evolution. Now, there was this necessity, as I mentioned, to evolve a machinery which would be removing those non-coding segments and getting rid of them in order to produce functional proteins. So I managed to tell you how it happened that we have them, but now, why we have them, but now how does it happen that they get removed? So this is a process which is known as pre-mRNA splicing. And in a very brief summary, pre-mRNA splicing is a process during which those non-coding introns, as you see here, are removed from pre-mRNAs. And that happens in two very simple reactions, during which one of the exons is reacting with the middle part of the intron and so on. And these are just two very simple trans-esterification reactions. So from the chemical point of view, there's nothing really complicated. But what is difficult is really to identify those elements which are coding and non-coding. And for that reason, there's this enormously complex machinery which is involved in excising them in human cells. And this machinery is known as the spliceosome. So this is very large and dynamic complex. And I know that probably for most of you, this is not of an interest, like very nitty details of the cycle. But I'm showing you a slide to emphasize that this is a very dynamic process which requires multiple different RNA protein complexes to assemble with those products of the genes in order to get rid of the non-coding message. Okay, so yeah, the cycle can be divided into multiple stages. Anyway, so what we are doing in my lab and what I was doing during my PhD and postdoctoral time is we're trying to understand the mechanism of this process which I just described to you by means of structural biology. So by looking at how the molecules look like, we want to understand their function and then how basically it happens in the cells. And if we think about structural biology, of course, the first thing which comes to mind is X-ray crystallography. So traditionally, X-ray crystallography was the only method that would allow us to look very deep into magnify biological molecules to the level that we could see individual atoms and so on, so that we could really understand the chemistry of life. And in a typical X-ray crystallography experiment, we need to purify proteins. So we extract them from various tissues or from overexpressing bacteria. We really need to make them very clean so that they can form crystals. So we have ways of putting very complex biological molecules together so that they form crystal, which is not made of salt like the crystals that you normally know, but this is entirely made of protein and solvents. So it really means that all the molecules are perfectly aligned and they form three-dimensional lattice. And if we have lattice like that, we can do X-ray diffraction experiment in which we record diffraction patterns, and then from that one can actually, using computational method, tell what was the structure of the crystal. And in our case, the crystal contains only protein molecules, so but at the same time, we can actually tell what is the structure of molecules which are forming the crystal. And that leads to the formation of sort of like a electron density map into which we build atomic models and interpret them. So these days, normally these kind of experiments are done on the very big machines known as synchrotrons. So this is one of the kind of particle accelerator, and that's placed in Grenoble. So that's a European synchrotron. And so this is actually where I'm working. So EMBL Grenoble is located right in between this nuclear reactor and the synchrotron source. Okay, so these are a couple of examples what we can actually learn from X-ray diffraction experiments on biological molecules. So this is something which I was doing during my PhD. That's the structure of one of the proteins, which is very important for the process which I was describing and for the pre-mRNA splicing. And just for those of you who really don't know much about structural biology, so all those different ribbons and strands they represent, these are just simplified representations of atomic models which we build based on experimental density maps. So we know this is not really how the protein look like. There are individual atoms of carbon, nitrogen, and oxygen. However, this makes it much simpler to understand, you know, what is really happening. So this is another example of the project which was carried out in my former lab by my supervisor Kiyoshi Nagai, and this was really like a flagship project of the lab, which was showing that we could solve the structures of all individual components of this machinery, and that's actually all those proteins are important for the very early stages of the splicing process, where the intron and exons are defined. But the reason I'm showing this, and that led to the structure determination of the entire particle. But the reason I want to show you this is to make you realize that these processes of forming crystals from proteins and making diffraction experiments are very time consuming. So in the case of the U1 snare, which you just saw, I mean, the entire project took the whole lab about 25 years to establish high resolution structure of this assembly. For the other thing which I was showing you, this PRP8 structure, we got a bit better. It took about 10 years for the entire lab and several generations of students and postdocs. But at that time, the goal of the lab was really to understand how the entire machine works. And it is about order of magnitude bigger. So it was pretty clear that it was very difficult to imagine, you know, how we were going to get there and in what sort of time frame. And it was clear that some kind of different approach was needed. And really what happened around the time that we were working on that was so-called resolution revolution in cryo-EM. So basically, different methods showed up which could compete with X-ray crystallography in determining the atomic details of biological molecules. And basically this method spread very quickly, and you probably have seen like quite a few review articles about that. And it was called very quickly like a revolutionary method. And this method is really called electron microscopy. So it doesn't really differ very much from the classical electron microscopy that you would probably learn about in school. In this case, we are using cryo-electron microscopy, which means that specimens are kept at very low temperatures. And in a typical cryo-EM experiment, one would purify those samples, the same kind of samples that I was showing you before. But there's no need of forming crystals, which is a bottleneck of the whole process. And in this case, we place those samples on the small 3 millimeter cryo-EM grids, and then remove excess of the sample and plunge freeze it in the liquid ethane. And the goal of cryo-EM experiment is really to form a thin layer of molecules, about 1 molecule in width, of the molecules suspended in amorphous ice, which is not as crystalline ice. And this kind of samples we insert into a microscope and record images, and from that we can calculate the structure. So, because in this approach we are looking at the molecules in the solution, there's no need to form crystals. Somehow it was revolutionizing for the structural biology, and it really becomes the method of choice at the moment. Okay, so this kind of data is collected on the electron microscopes, and this is like an example of how the electron microscope look like. And it's one of the very powerful ones, where the electrons are generated at the very top of the machine, and they are accelerated throughout the column, and then they are passing through the sample, and then they are imaged on the detector. And the fast electrons from my title, if you're wondering what is it about, it's really, if you think about there's the potential difference inside the microscope is about 300,000 volts. So those electrons which are generated then, they achieve nearly a speed of light when they are passing the sample specimen. Okay, so if you are wondering what is this peculiar background on my first slide, this is kind of image that we are getting from an electron microscope. So this is one of the images which I used to reconstruct three-dimensional structure of one of the proteins I was working with. And if you have good eyes, you can probably see that there are some darker spots which are individual particles of biological molecules. Of course, I didn't really mark all of them. So one thing which you can probably see here that, this doesn't look particularly convincing. I mean, you see that some densities are a bit darker than others, but you really cannot tell any details about this. This really look like a bit darker spot in the micrograph. And that's true because the signal-to-noise ratio in electron micrographs are very low because the specimens are very radiation sensitive. So the way we deal with this data, we are typically picking hundreds and hundreds of thousands of those particles, and then we start averaging them. And once we start averaging them, we can boost signal-to-noise ratio, and then you start seeing more details. So these images of those particles are generated from multiple different particles averaged together in the same orientations. And from those two-dimensional images of the specimens, we can, using some computational methods, we can generate three-dimensional structures. So there are various computational methods which are allowed to back project and restore three-dimensional object from that. And those objects have to be interpreted because at this stage, it's really, that map does not really tell us very much about the biology. So we need to build atomic models into those maps. So this is basically represents some of the RNA molecules and some proteins. And once we build atomic models, once we interpret the map, we can then start asking questions, what does it tell us about the biology? And this is the last part of my talk. I want to tell you what we have learned from some of those structures that we solved. So this is again the splicing cycle, which I showed at the very beginning. So it is quite complicated, but during the last maybe 3 or 4 years, we and also other groups, we managed to solve multiple different structures of those assembly intermediates. And in this case, it is fair to say that the molecular movie started emerging. So we have different frames of the movie, and we can start really observing how those processes happen at the molecular level, and step by step follow what is really happening. And I just want to show you a couple of examples of the things which we were particularly interested in, and mainly catalysis process of the splicing. So this is kind of a model that we obtained from one of those maps, and you can see that there are many different proteins and which are represented by those different tubes and surface representations. And yeah, without going into any details, because I understand it's not probably of great interest, we managed to have a very clear insight into the active side of this big machine. And the active side of the spliceosome is based entirely of RNA. So these are 5 RNA molecules which are coming together, and then they allow splicing catalysis to happen. So that was one of the very big insights from this structure. And but as I mentioned, we like to look at the movie, so we like to look at various different consecutive steps in the process. And we know that the splicing has 2 steps, and we are looking in quite a lot of details into how spliceosome is undergoing transition from first to second step of chemistry. So here you can see, there is quite big conformational change of the entire machine when it goes from one step to another. And then when we look at the active side, it explains why it has to happen. So the active side of the spliceosome is very tightly packed, and on the molecular level, what happens when you saw this very big change? One of the first step components is getting removed from the active side. And this makes space for the second step components to come in, and they can form the active side for the second step of chemistry. Okay, so I think I'm going towards the end. So the conclusion from this part of my talk is really that those newly emerging cryo-electron microscopy methodologies allowed us to solve the structure of the spliceosome and really to understand the process in great detail. And I think that sort of like a bigger message is that to understand biology at the molecular level, we really need structural information. And we have nowadays, we have methods which allow us to do this and to investigate processes in great details. Okay, so with that, I want to acknowledge everyone who was involved in this project from my former lab, particularly my former mentor Kiyoshi Nagai and people from his group, and my own group at the EMBL Grenoble, where I'm working at the moment. And I will just use this opportunity to introduce EMBL to you. So EMBL is the laboratory where I work, and this is a laboratory which is formed as the intergovernmental agreement between several different countries. And at the moment, we have 6 different locations. About 1,700 people work at the EMBL, about 80 nationalities. So we have the main laboratory, which is based in Heidelberg. And we have also outstations in Kingston, Grenoble, Barcelona, Rome, and Hamburg. And the mission of EMBL is to do basic research and to promote molecular biology in Europe. And we're also providing a lot of services, advanced training, technology transfer, and so on. So yeah, I would encourage you to have a look at the EMBL websites, and then if someone is actually looking into possibilities of doing work with us, there are possibilities for various stages of researchers, from PhD student, postdocs to group leaders. And so with that, I would like to thank you for your attention. I'm happy to take questions.